activating a behavior. So when I look at the surface of the cell, actually, instead of being smooth, there are all these structures like antennas sticking up all over the surface and proteins built into the membrane. And that these proteins read the environment and convert that environmental signal into a behavior. Let me explain how it works. Here's the membrane. Here are two different proteins. I'm going to tell you right now, there are thousands and thousands of proteins in the cell membrane, but I can divide them into two groups. One group has antennas on it. And antennas are receivers, just like your television antenna on top of your house before cable. When you had an antenna on the top, what was its function? To pick up a signal. And then what happened to that signal? It was transmitted down the wire to the television, and the television converted the signal into something you could see. Well, here's the point. Many of the receptors in the cell have these antennas sticking up from the cell. So if I go back, these are the antennas sticking up from the surface of the cell right here. And what they're tuned to are not television stations, they're tuned to environmental information. It might be glucose, for example. Is there sugar out there? Or histamine. Is there histamine out there which tells me to get ready for an emergency response? Or is there something like insulin which tells me to change my metabolic pathway? For every different thing the cell can see, it has a different antenna. So that means the cells are covered over the surface with antennas for everything the cell can deal with. So the signals come in and picked up by the antenna, but then they're converted into the behavior by the second class of proteins. There are three different kinds. Channels, which are just like olives with a hole in the middle of them where information can go in. Enzymes, which are proteins that cause metabolism to occur. Or cytoskeletal proteins, proteins that change the shape of the cell. So if a signal comes in, let's say a toxic signal comes in, and this is a receiver for that, and it couples to the skeleton, it says, toxic signal, turn around and start running the other way, then this is the input and this is the output. So the proteins work in combination. Receptors receive signals. Do you have receptors? Yeah. What are the obvious ones? Eyes, ears, nose, taste, touch. Where are all your receptors located? In the skin. So you and the cell are parallels, but it doesn't look like an eye and it doesn't look like a nose, but its function is exactly the same. It's the equivalent of an eye. If this was tuned to light, a photon of light would hit this antenna and that cell would respond like an eye and say, I see the light. And once it sees the light, it has to convert that information, and it uses it by connecting to this, and I'm going to show you how that happens. So the input, the antenna, connects to the output, which makes the behavior. Let me explain how it works. So we'll use this model right here of a receptor. Let's see. This receptor is going to, I'm going to show it to you in dynamic motion. Here's my membrane, bread and butter sandwich. Here's my antenna, and this is the antenna scouring the environment for a signal. Look at the shape, because the protein goes into the cell. Look at the shape. What happens when when a protein binds to a signal? Changes shape. Well, watch what happens. See, when the signal came in, and watch what happens when the signal goes away. Should I do that one more time so you can see that? Okay, basically it says this, is that the proteins are in the cell, that these receptors are migrating through the surface of the cell. Okay, here it is. Are you going to come start moving soon? Okay, here's the protein. It's scanning the environment, looking for signals. And let's say it's an insulin receptor. If there's insulin, I would activate the receptor. But look, no activation of the receptor. There's no change in shape. But when the insulin shows up, I change the shape. So if I'm inside the cell, if we were inside the cell and these proteins were hanging down from the ceiling, would you be able to tell if insulin was here or not by the shape of the protein? Yeah. So if you're inside the cell, you can tell what's going on outside. Okay? Now the next thing is this. After I receive a signal, I actually have to send a secondary signal into the cell to activate the behavior of the cell. So in this case, I'm going to use a channel, which is like an olive. And what happens is this. This protein moves around through the membrane. See, it's closed at the top. Nothing can get through. But when it opens, it makes a channel where signal molecules can shoot inside the cell. And the bottom line is this. The only thing that can go through this channel is what fits. If the molecule doesn't fit, it doesn't go through the channel. So basically it says this. In the resting state, the protein is closed. But when activated, the protein opens. And when it opens, it allows signals to go into the cell and coordinate the behavior of the cell. So now that we've looked at that, let's put the two together and then show you what is this mechanism that controls biology. The two together, the input and the output. This is looking for the environmental signal, and this is going to convert that signal into behavior. This is the connector. Look at the shape. Does it fit here, yes or no? Okay, so that means no signal is present. So no behavior. But when the signal shows up, I change the shape. When the shape is changed, then I connect it, open this one up, and see this? This is the signal that tells the cell to do something, and it's going to do whatever is connected to that. So the bottom line is the behavior of the cell is controlled by the input of the environment, and then the conversion of that input into a behavior signal that coordinates the function of the cell to respond to what's going on in the environment. Interesting point. If I cut the antenna off the cell so it cannot see anything, there's no behavior. The cell is totally comatose. It'll sit there, and what does that mean? It says then behavior is related to the signal. If there's no signal, there's no behavior. So it's like reflex, stimulus, response. The signal is the stimulus, and then the cell creates a response. So basically it goes like this. The receptor is the input. It reads the environment. The receptor then connects to the effector, which generates the behavior through a secondary signal. The primary signal, this is the secondary signal that activates the behavior of the cell. So that this unit is controlling a specific aspect of behavior. But there's like thousands of these different kinds of units simultaneously. So this is an example of just a switch. It's a switch. When insulin shows up, switch on metabolism. When histamine shows up, switch on protection. So for each different thing that's out in the environment, there's a switch that will activate the behavior of the cell. So basically, did you understand that as a switch, input-output? Well, then look at it this way. The function of the receptor is awareness of the environment, right? It sees. But I have to convert that signal into a biological action. So the function of the output guy, the effector, is to create a physical sensation or response to that signal. Is that understandable so far? Why is this important? Go back and ask, the first step is this. What does this do? What does it do? It controls a typical behavior in response to the signal. Well, what is this called? Well, it represents awareness of the environment through physical sensation. So what is this control unit actually called? Perception. Perception, awareness of the environment through physical sensation. So if I go back a second, I say, what is the function of this? And the answer is this. It's perception. It sees the environment and activates a behavior. There's no DNA involved. There's no genes involved. All we're talking about is stimulus-response. Stimulus comes in the receptor, response made by the effector. Okay? So the point about it is this simple first conclusion. It's very basic. Behavior, which is movement, this is behavior. Movement of protein is controlled by the signal, but via perception. So the bottom line is this. Perception controls behavior. If there is no perception, there's no behavior. And all of a sudden it's like, oh, well, that's number one. We didn't have genes involved yet. So let's just see how this actually works. So basically, here are proteins in the cell. Let's say that these proteins carry out a specific function, respiration or digestion. We call it a pathway. So let's say it's a muscle contraction. That these proteins, when activated, what activates the protein? The signal activates the protein. So what we're going to do is convert an environmental signal using the receptor and the effector. And this time I'm using an enzyme. Okay? And what I'm going to show you is how the mechanism from the environment, the signal, well, here's my connector one. Remember the green guy? But now the signal comes, and when the signal from the environment binds to the receptor, it changes the shape or conformation of the receptor, which implies that the signal is there. So the protein informs the cell that something has happened. Now that that shape has changed, this processor protein, the green one, can bind to the shape because it wasn't able to bind before. It wasn't the right shape. It's like dominoes, one hitting the next. Now that the processor protein is bound to the receptor, it changes its shape and will conform to fit the enzyme. So one domino hits the next, hits the next. Now that this is connected, I activate the enzyme, and the enzyme is going to create a signal. Now here's the connection between my muscle proteins and the receptor. This is the secondary signal. Remember, primary signal and secondary signal. Well, at first it's bound and covered up and inhibited because if there's no signal, I don't want it to do anything. But when the